So in lecture, you should have been talking about cellular transport and the difference between a passive transport and an active transport. So we're going to talk specifically in lab about osmosis, osmosis being the movement of water. So you should have received this note page, and so you can pause the video at any point in time and write these vocabulary words down. They're also on the PowerPoint that's up for you. Okay, so this is the note sheet that you should have. So let's start. So first I need you to understand the difference between the word solute and the word solvent. Solute is the substance that's being dissolved in the solution, being dissolved by the solvent. An example would be sugar. Sugar would be a solute. An example of a solvent would be water. So if you take sugar and dissolve it in water, you get a solution, and a solution is the two put together. So the way you can remember, because students do mix these up, solute is the smaller word, and the stuff that you have less of, like sugar, would always go in the thing that you got more of, like water. The other way you can remember is this got a U, and so does the word sugar. So you know that sugar dissolves. All right, so on we go. So you should be talking in lecture class about diffusion. Diffusion is a passive transport, and it's a way a lot of times our cells can get their nutrients. And this is always the movement of from high to low concentration. Molecules are going to be moving from high to low, such as if you were to spray perfume, it would be concentrated in one area, eventually it would spread out. If you were to take some dye and put it in water, eventually it would evenly spread out. If you take sugar, a lump of sugar, eventually over time it's going to spread out and get even. And so there's no energy required for simple diffusion. And so this is our lipid bilayer, and you're not seeing the proteins in here. And so some of the molecules are small enough that they can just pass between the lipid bilayer. They don't need the protein channel to go through, so they can just easily diffuse through the channel. So this is a simulation just to kind of show you if we had a membrane here, and if we had a bunch of solute on one side and not a lot of solute on the other, there are times where the molecules, if they could, they would, these little yellow dots would like to get through over here and even it out. That's the whole point, is to even it out, to diffuse. But there are times where this molecule is too big and it will not fit through there. And so what we'll see happen, in order for the concentration to equal out, we will see the movement of water. So we're going to see water, and water is always going to go to the side that has more solute. We have terms for this. If it's got more solute, this is called hypertonic. Hypertonic is a solution that has more solute. The side that has less solute is called hypotonic. So hypotonic, hypo means low, low in solutes. Hyper means high in solutes. We're talking about the stuff. And what we're going to see is water will always pass from the hypo side to wherever there is more solute. There's a thing that you'll learn in AMP2 when you talk about your kidneys, etc., when you're doing the excretory system, is that water always follows the salt. So that's kind of a thing. So water is always trying to basically dilute. Think about, if your tea is too sweet, what do you need to do? You got to add water. So if this was sugar over here, we're too sweet on this side, so water would have to move over here if the sugar were not allowed to come over to the other side, if we had that selectively permeable membrane. So just remember, it always leaves the water always leaves the hypotonic side and goes to where there's more stuff. That way you can remember what's going on. So the definition of the movement of water or diffusion of water is osmosis. This is a passive transport. So is diffusion. Diffusion is just any old molecule. This is specifically water movement. So it doesn't require any energy, it's just going to move on through. It is a passive transport. Wrong direction. So hyper always means high. Hypo always means low. Hypoglycemic, hyperglycemic, high in sugar, low in sugar. Iso, like an isosceles triangle, is going to mean equal. And then it's talking about, we see the word tonic, that means tension, and it's the tension that's being caused by the solute. So if this is our cell, do you see this has got a lot of stuff? This would be hypertonic, this would be hypotonic, and this would be isotonic. If we count the little green dots, they're the same on both sides. And normally we talk about solutions on the solution outside the cell, so the extracellular fluid is usually how we term that. So if you want to pause the video, we just said that solution with greater amount of solutes, solution with lower amount of solutes, solution with equal amount of solutes. You can pause the video and get those vocabulary words. So let's look at this little activity. I'll be pausing the video, but so you can see we've got 50% solute. So what I'm going to do is draw 5 dots, 2, 3, 4, 5, and then 50. I don't want to draw 50, so I'm just draw 5. And so here I'm going to draw 7. So you can see here I'm going to draw 4 and 4, inside, outside, 6 and 2. So I'm going to pause the video if you want to do that. All right, now that I've got my dots up here, if you'll notice I've got 5 dots here on the outside, 50% solute. We've got 7 in here. So we've got a higher amount in here. So this outside here is a hypotonic solution on the outside. On the inside, it is hyper, okay, but we really care about the outside of the cell, especially in lab. We're always going to be asking about the outside of the cell here. It is hyper. Hyper means it's got more. So what's going to happen to the water? Where's the water going to go? If you have a hypotonic solution that you've just stuck your cell into, so the cell always or water will always move in the direction where there's more stuff. So water is actually going to go inside this cell. So water goes into cell. So what's going to happen to that cell if water starts going into it? Well, that cell is going to get bigger, right? So the cell will swell, get bigger, gain mass, etc. There could be a point where the cell gains so much that it might burst, and it might burst. If it bursts, that's called lysis. If it's a blood cell, we call it hemolysis. Hem means blood, red blood, and so hemolysis is the bursting of your red blood cells. Does that make sense? Because water has gone into that. All right, let's look over. Let's do the one at the bottom. So we have 60% on the outside, 20% on the inside. So we have a hypertonic solution. Okay, because it's higher on the outside. If you take a cell and put it in a hypertonic environment, water is going to go out. That's where water is going to go. So I meant to put water on here. Water went in, water went out. So water is going to go out of the cell. What's going to happen to the cell? Well, the cell will shrink, shrivel up. Shrink, kind of crinkles up, will shrink, shrivel, whatever word you want. When the cell shrivels up, that is called crenation. Crenation is the shriveling of your cell, or crinkling if you want to say. Not good. So if you've ever been washing dishes and for a long time and you have your hands in the water and all of a sudden it looks pruny, that's what's happened. Your cells have kind of shriveled up. All right, here we've got 4 on the inside and 4 on the outside. So we are in an isotonic environment. So it looks like nothing's happening with the water for isotonic because it is equal. But honestly, water is going inside and out at the same rate. So it goes, water's always moving, goes in and out at same rate. So the cell will almost stays the same. So stays same, almost stays same, just very little change for an ISO. This is the environment we want our cells to be in. All right. So here is a prettier picture of that. So you can see if we put our red blood cells in a hypotonic solution, if it goes in too much, swells too big, it will lyse, it will destroy itself. This is showing water going in and out, which is an isotonic environment. And this is water going completely out of the cell, and that means we've put it in a hypertonic solution, and that's causing that crenation. So there is the vocabulary for that. You can pause the video and get that. So this lysis means generic. Hemolysis is specifically about the red blood cells because this heme means red blood cells because of the iron group that's in your blood. So you can see the picture here. Here in a normal isotonic, if we put it in a hypotonic, it's going to swell, water's going to go in, could lyse, or if we put it in a hypertonic, it's going to crinkle up. So if we look at this cell membrane, here we've got that lipid bilayer. Remember, we've got the head and we've got the tail, and we use those terms that the heads are hydrophobic or philic. They're towards the water. Can you see that? So these are hydrophilic because they're water loving, and these tails are hydrophobic. So we're looking at that. So I wanted to point out this PowerPoint or here on this slide. We think when I was in school, we used to believe that water just went straight on through this lipid bilayer. We now know that there's actually a special protein for water. It stays pretty much open, but it just only allows water to go through, and that's called an aquaporin. And for lecture, I definitely want you to know aquaporin, special protein for the water movement for osmosis. It is still passive transport, agua meaning water, porin on through. Okay, so I'd like you to know that, especially for lecture, special protein for this osmosis. All right, that concludes osmosis. See you soon.